Good morning, everyone. My name is Masahiro Ito from Toyo University. First, I would like to thank to the organizing committee for giving me this opportunity to present our current work in this session. Today, I will talk about our research entitled Alkylphilic Strain A5 facilitates mass reduction and amino acid release from soy sauce Moromicas. Let me begin with the global issue of food waste. As you know, food waste is a serious problem worldwide and is closely linked to SDGs Goal 12, responsible consumption and production. According to a recent report from UNEP, about 1 billion tons of food waste were produced globally in 2022. This is an extremely large amount. So in Japan, we also waste a lot of food every year, but where does this food waste come from? As you can see in this table, about 60% of food waste in Japan come from food manufacturing process. So around 10% of food loss is made up of leftover from fermentation and extraction. So finding a way to reuse these food residue is really important, and it is something we need to start working on now. Our specific target in this study is soy sauce Moromicas, also known as a soy sauce press cake. In simple terms, Moromicas is a solid residue left after soy sauce is pressed. In Japan alone, about 60 kilotons of Moromicas are generated every year. At present, most of it is used only for low-value purposes, such as animal feed or fertilizer, or simply throw away as industrial waste. But why is it so difficult to recycle Moromicas more effectively? There are two main reasons. So first, it has very high moisture content, which make it heavy and easy to spoil. Second, and more importantly, it contains a very high salt concentration, often around 15 to 18%. This high salinity is toxic to most conventional microorganisms. As a result, standard biological treatment such as composting or methane fermentation are extremely difficult to apply. Let me briefly explain how soy sauce is made. Soybean and wheat are fermented with koji mold and salt water to form moromi, which is aged for several months. After that, moromi is pressed. The liquid becomes soy sauce, and solid residue is moromicas. So our research question is quite straightforward. Can we use biology to handle this salinity wet and difficult to treat residue in a particular way? To address this challenge, we focused on alkalic bacteria, which we called alkalophile, which grow well under high pH conditions. We choose this strategy for a very practical reason. In general, biological treatment of organic waste requires strict sterilization to prevent contamination by bacteria and fungi. However, sterilization is expensive and uses a lot of energy. In contrast, but when we keep the pH high above 9, the growth of most common contaminants is naturally suppressed. This allows us to operate a low-cost, open, non-sterile fermentation system without strict sterilization or expensive antibiotics. This is a key advantage of our approach. This slide shows the overall roadmap of our project. Our goal goes beyond simple waste treatment. We aim to turn waste into something useful. The process has three main steps. First is the construction where alkaphic bacteria break down solid Moromycas. Second is platform building, where we obtain an amino acid-rich liquid. Third is upcycling, where this liquid is used to make high-barrier products such as bioplastic or ectoine. Today, I will focus on the first step, how we isolated effective bacteria and tested their ability to degrade Moromycas. Let me now explain our screening process. Our aim was to identify bacteria that can survive under both alkaline and high salt conditions. To do this, we collected samples from different locations, including soil from around our university campus and the unique environment of Utsuno Cave. From these samples, we isolated 21 alkaphic strains. For the primary screening, each strain was inoculated into test tube containing alkali Moromycas medium and incubated. We then simply checked by visual inspection whether the solid Moromycas particles were solubilized. Let's take a look at the visual result. The tube on the far right is the control, and as you can see, it is still cloudy with a lot of brown solid materials. By contrast, the tube in the center, strain A5, look much clearer. This visible difference suggests that strain A5 is particularly effective in solubilizing solid Moromycas. We next quantified what we observed visually. We looked at the residual solution percentage, which tells us how much solid material remained after cultivation. For many strains, between 60 and 90% of the solid remained, suggesting limited degradation. In contrast, strain A5 reduced the residual solid to about 20%, meaning that nearly 80% of the solid Moromycas was degraded. For this reason, we selected strain A5 for further analysis. Next, we wanted to understand why strain A5 is so effective. To answer this question, we analyzed the extracellular enzyme secreted by the bacteria. The graph on this slide shows protease activity on the left and amino acid activity on the right. Here, I would like you to focus on the protease activity. The orange line represented strain A5. As you can see, compared with other reference strain A5 showed the highest and most stable protease activity under alkaline condition. This strong protease activity is likely a key factor in the efficient degradation of protein-rich Moromycas. This motivated a cell-free enzyme-based degradation approach. This slide summarizes the results of the cell-free system. Solid fraction is steadily reduced over time, reaching a reduction of about 6 to 4%. In parallel, the concentration of free amino acid increased, reaching over 200 millimolar. Taken together, these results demonstrate simultaneous waste reduction and amino acid production. The next challenge is the amino acid disappeared quickly. The question is how we can stabilize them. Next, we examine whether we could further improve production efficiently. At the standard substrate concentration of 1%, we suspected that highly activity enzyme in the supernatant might not be fully utilized. To test this, we performed a substrate loading experiment, increasing the amount of monomicas from 1 to 3 grams per 100 milliliter. As shown in this bar graph, total amino acid concentration increased as the substrate loading increased. These results indicated that a higher substrate loading leads to higher amino acid production, allowing us better exploit the enzymatic capacity of the system. Of course, producing more amino acid is not only goal. We also have to make sure that the waste is still being broken down efficiently. This graph shows that even when we increase the substrate loading, degradation process smoothly. So we are not forced to choose between production and waste reduction. We can achieve both simultaneously. Next, I'd like to explain how we made better use of the amino acid produced in this process. During the reaction, we noticed that the amino acid concentration did not always keep increasing. In some cases, it even started to decrease. When we examined the system more closely, we found that by day 3, the amount of remaining protein was almost zero. This suggested that protein degradation process efficiently and at an early stage, but the free amino acid can later be affected by other enzymatic activities. To address this, we introduced a simple heat treatment step to suppress amino acid degradation enzyme. As shown here, applying heat 1 day after added the A5 supernatant was almost effective timing. This simple approach helps preserve the amino acid produced and improves the overall amino acid yield, leading the effective use of these amino acids. This slide shows that the time course of the total amino acid content. When we add 5% of the A5 culture supernatant and applied heat treatment on day 1, without heat treatment, the amino acid leaves gradually decreased over time. In contrast, when heat is applied on day 1, amino acid concentration is stable, maintained. This is an important practical point because it makes the amino acid rich solution much easier to store, transport, and handle. Overall, these results show that our method enables high variable utilization of food waste with really industrial application. In this study, we successfully isolated A5 strain from soil samples. We identified this strain specifically for its ability to degrade soy sauce moromicas. When we evaluated its performance, the A5 strain showed superior mass reduction compared to other strains, achieving a residual rate of 20%, the key to this successful line in its enzyme. The A5 strain demonstrates high activity in both protease and amylase. These enzymes break down the protein and polysaccharides in the waste, which is what enabled this significant mass reduction. Next, let's look at the result of adding A5 culture supernatant. By adding just 5% of cell-free supernatant to the medium, we achieved two goals simultaneously, reducing the mass of soy sauce moromic acid and producing amino acid. We also found that increasing the amount of moromic acid in the medium led to a further increase in amino acid production. So finally, we approached the heat treatment to the medium. This confirmed that the produced amino acid can be successfully maintained. This stability suggested a strong possibility for effective utilization, such as functional food and supplements. Looking ahead, our main goal is to turn soy sauce waste into valuable resources. To do this, we have three steps. First, we want to produce more amino acid. Second, we will check exactly what kind of amino acid are made. And third, we plan to make our system bigger for factory use. We see two main use for this research. One is for food. We can use the amino acid for healthy food and supplements. The other use is making material like bioethanol and bioplastic. We can use the waste broken down by A5 to feed other bacteria. By doing this, we can turn waste into something new and useful. So last, I would like to express my sincere thanks to the Shoda Shoyu Corporation for generously providing a Moromicasu sample. I will also thank to all members of our laboratory for their hard work and support throughout this study. Thank you very much for your attention. I'll be happy to talk any question.